Welcome! In this video we demonstrate Mu-8, an interactive visualization technique for analyzing differences between a mutated protein sequence and its family. The question we try to answer is, which mutation did cause a protein to lose its function? At the heart of Mu-8 is the sequence view, which lists all amino acids that make up the mutated protein. Red amino acids are mutated, while white amino acids are identical to the original sequence. Above and below the sequence, we stack bars that show how different the amino acids are compared to a large family of functional proteins. We show bars for each of the 6 characteristics that describe the important biochemical properties of amino acids. The height of these bars is based on a score that measures how different the amino acids are for each characteristic when comparing it to the family. How these scores are distributed are shown in the 6 histograms at the top. You can use these histograms to filter the scores that are rendered onto the sequence. For example, I can remove the tails of the alpha helix and turn propensity. I can completely remove it. I can completely remove all scores, select all. Each interaction with the histograms filters the scores that are rendered onto the sequence. Below the context sequence, we see the focused view. This focus view makes it easier to read the details for the brushed region of the context sequence. In this view, we can do several things. We can switch between stacked and aligned bars. This provides another lens to compare bars both within and amongst residues. You can also see what the original residues were in mutated positions. Which part of the sequence is enlarged is driven by the selection window in the main sequence view. You see that we can move the selection, completely brush other regions, and transition between stacked and aligned views. In between the context view and the zoom, we show a conservation heat map. This heat map encodes the variation of scores amongst the family at that position. The default view is black and white, and when the user brushes a region, the colors are encoded. Blue corresponds to alpha helix and turn propensity in the first row, and so on. Using this encoding, the user can identify regions that have low variation and high variation. "3D structure and position is important for proteins. We take this into account in 2 ways. First, we have a distance histogram on the right, and on the left we have the full 3-dimensional structure of the protein. Using the distance histogram, the user can filter the distances from the selected context region to other residues on the sequence. Dark chords are closer, and lighter chords are further away. Furthermore, we include a 3D view. This 3D view initially auto-rotates, but you can stop this. You can turn the 3-dimensional structure, zoom in, and out, and the highlighted region will change as you change the brush on the context sequence. The data used in this video shows the mutated dysfunctional protein D-TIM and the differences to SC-TIM and its family. Using Mu-8, we can see that several regions are quite different from the family, especially the amino acids at positions 150 to 156, shown here. This region has three properties of interest. First of all, you can see all of the outliers, both significantly greater than the family and significantly less than the family, across this region. Furthermore, you'll see that for most of the region, the positions are highly conserved amongst the family. Lastly, using the distance histogram, you can see that this region is also connected to other regions that have outliers and that are conserved. That concludes the demo, and thank you for watching the video.